Assalamualaikum warahmatullahi wabarakatuh, everyone. Thanks again. Today we will look at the viral pathogens and associated diseases. Just a few reminders. If you watch the video, please click on your I attend or your attendance in my personal web pages so that I know who are watching the video. So this kind of a replacement class in the form of asynchronous virtual lecture. I hope you can gain some benefits from this lecture. This is viral pathogens and associated diseases. We are going to learn about the viruses and some of the disease issues associated with the pathogens. Okay, so for the objective, we can describe the general properties of the viruses and also explain the replications of these two types of viruses, which are the RNA and also the DNA viruses. I hope you can outline the classification of medically important viruses and some lab diagnosis to detect these viral infections. So what are the viruses? Okay, what are the viruses? The virus is a very small living particles that can infect cells and change how the cells functions. Okay, once you got a viral infection at the cellular level, okay, you can find that these can change the activity of the host cells. How about the virions? The virions is a complete virus particle which is in the form of DNA or RNA core and also the protein coat as it exits outside the cells. This is the viral particles. Whatever it is, okay, it's originated from the virus itself, whether it's RNA or it's DNA, it's the capsid or some other protein structure. This is also called as the infectious viral particles. How about the virions? Virions is nucleic acid without protein coat on its own, okay, so we call it virion. Like the DNA only or RNA only. Some viruses they don't have this coat, so we call it virions. Small infectious agents of plants. Okay, prions. This is a type of disease causing agent that is neither bacteria nor fungal nor viral. Okay, and contains no genetic material. It's the disease. Okay, it's not the particle from the virus or bacteria or any other fungus. General characteristic of virus. We know that the virus are of many different types, and you know that the classification of virus can vary. And most of the time we consider this virus as an obligate intracellular parasite. What does it mean? Obligate intracellular parasite means that these virus they cannot reproduce outside their host cell, which means that the parasites reproduce or the reproduction is entirely relied on the intracellular resources. Okay, it depends on the host cells to give the nutrients, to give the energy for them to carry out with the replication processes. The virus contains a single type of nucleic acid in the form of DNA or RNA. It contains a protein coat. Sometimes it's self-enclosed by an envelope of lipids, glycoproteins, proteins, or carbohydrates that surround the outer structure of the nucleic acid. Okay, we call it this as a capsid. Okay, capsule, like a capsule, so it's protecting the DNA or RNA or the genetic material of the virus. The virus multiply inside the living cells by using the synthesizing machinery of the cells, means they are manipulating the ready-made system inside the host cells, and they try to hijack this system by making or altering this genetic information, and they cause the synthesis of specialized structure that can transfer the viral nucleic acid to other cells. So it's kind of manipulating the host cells, and after that they try to make the another copy of their own viral cells. The host range. What do we mean by the host range? Okay, the host range refers to the spectrum of host cells the virus can infect. Okay, this example of the Bacteriophage. I think this is T4 Bacteriophage, a virus that commonly infect the bacteria like the E. coli. It has a very like a robot structure. The bacteriophage or simply phages are viruses that infect bacteria. Host range of a virus is determined by the virus requirement for its specific attachment to the host cells and the availability within the potential host of cellular factors required for viral multiplication. In other words, the viruses are very selective in terms of choosing the range of host cells that they want to infect. Okay, there are certain materials that they can attract the virus to bind to. Some viruses, they don't have the susceptibility or the opportunity to bind to the host cells due to different structure or different nutrients that is present on the host cells. According to the science, if you look at the morphology, we can see that the scientists determine the viral structure in comparison with some other small particles or small pathogens. They did it using the electron microscopy. If you talk about virus, commonly it refers to the particle size ranging from 20 to 1000 nanometer in lines. Okay, in lines. So you can see from the right hand side here, the human red blood cells, 10,000 nanometer in diameter. How big is that, right? You can see the plasma membrane of the red blood cells. It's around 10 nanometer thick. Okay, so you know that the virus is around 20 nanometer, and of course you can see how it's just like the double size of the plasma membrane of the RBC. You can see here the bacteriophage, which commonly infect the bacteria, is around 225 in length. Okay, 225 nanometer in length. Okay, we are characterizing based on the length. Okay, how long is the virus? Chlamydia. So this is the chlamydia elementary body, around 300 nanometer. The chlamydia, usually it's the what they call as the common sexually transmitted disease. It is caused by the bacteria called the chlamydia trachomatis. It can infect both men and women. Okay, the women can get the chlamydia in the cervix, in the rectum, or in the throat, whereas the men can get the chlamydia in the urethra, which is inside the penis, rectum, or inside the throat. Right, so it's around 300 nanometer. But other than that, you can see the comparison. Okay, within this diagram, the images of the viruses, like the Ebola virus, is quite big and long. We can see the E. coli is in comparison. Okay, it's kind of like E. coli can eat all of these viruses, but it's not the case. All right, so this is just a side-by-side comparison to see how big and how small or how long the virus are. So these are the types of viruses that we are going to learn today. In terms of the structure of the morphology, the virion is a complete, fully developed infectious particle. If you get any of these viral particle in your blood system or in your tissue, it's quite difficult to get rid of them. Some of them can be infectious, some of them are not infectious, so we call them as a virion. It composed of a nucleic acid and surrounded by a protein coat. Viruses are classified by differences of these protein codes, right? So nucleic acid, of course you know about this nucleic acid. It can be in the form of deoxyribonucleic acid or the ribonucleic acid. Okay, the deoxyribonucleic acid or DNA, this is the double-stranded strand. Okay, the RNA on the other hand is only single-stranded. The virus can be single-stranded or double-stranded. It depends. It can be linear or circular in structure. The amount varies from a few thousand nucleotides to as many 250,000 nucleotides. So this is the amount of nucleotides inside the DNA or the RNA of the virus. Okay, so the classification of virus we can learn about this after. Okay, after this. Virus structure, again, the capsid and the envelope is also part of the virus structure. You know, the nucleic acid of a virus is surrounded by a protein coat. We call it as the capsid, and inside the capsid where we can find the genetic material. Each capsid is composed of protein subunits called the capsomeres. The arrangement of capsomeres is characteristic of a particular type of a virus. So this is the in terms of the morphology. Okay, based on the shape of the virus, you can classify them. If the capsomere, you know, is in the form of icosahedral or in the form of helix or helical, so you can know, or you can differentiate between these two type of viruses. Some viruses have capsid covered by an envelope, some don't have this one. Okay, some envelopes are covered by spikes. They are the carbohydrate plus the protein complex that projects from the surface of the envelope. They are used by virus to attach to the host cell. It's like a hook. It's like a glue that can attach. Okay, the spike is the thing, like a tool for the virus to attach to a specific linings of the tissue or cells, or it's like a tool or attachment, a tool for the virus to bind to. For the non-enveloped viruses, capsids are not covered by an envelope. Okay, it's just like a barrel like that, so they don't have any protecting material. The virus structure, you see the general morphology. The different morphological types are based on the capsid architecture. If the capsid, you know, are helical, okay, or polyhedral, so that is where we want to differentiate. Okay, that is how we can differentiate them based on the shape. The viral nucleic acid is found within a hollow cylindrical capsid that is helical structure, we call it. Okay, this is true for rabies and also Ebola virus. They are considered as the helical viruses. Whereas for the polyhedral, the capsid is in the shape of icosahedron. Okay, this in the shape of the icosahedron. This is true for the adenovirus and also polio virus. For the envelope virus, most of them, usually we can find them in a rounded circle structure or spherical. This is like the coronavirus, like the influenza virus or herpes simplex virus. Finally, we have another type of viruses which are complex viruses. Most of them are the bacterial viruses. This is just like the Bacteriophage that I have shown to you previously. They have capsids, and at some point they have this additional or extra structure attached to it. Example is like the pox virus, right? And also Bacteriophage. Most of the time we talk about the Bacteriophage invading the E. coli system. This is just an illustration to show you the different structure of capsomere arrangement that give rise to a different structure or shape of the virus, whether it's helical or it's icosahedral, whether it's an envelope or whether it's the bacteriophage or complex virus. The classification of DNA and also RNA virus can vary. For example, DNA here you can see DNA being classified based on the capsomeres arrangement, which is into the icosahedral, helical or complex. Further down icosahedral you can find the naked and also the envelope virus. And under the naked, you can see we can find the positive sense or negative sense virus. Okay, SS is single-stranded, DS is double-stranded, stranded secular, double-stranded linear. For the envelope, you have double-stranded circle gap, double-stranded linear. You have the naked envelope one, which is cytoplasmic origin. You have the double, or give rise to the double-stranded linear. In other words, there are different classification of different types of DNA virus based on the structure, based on the presence of the envelope, based on the single-stranded or double-stranded, based on the positive sense or negative sense, which we're going to explain and discuss further. Down here in the blue box, you can see like the Parvo virus, they are negative sense, and you can find around 18 to 26. I think this is bases, and this is the sequence, and also this is the amount of protein. I think this is the amino acid, okay, that is being translated from this mRNA or from this sense, what we call the arrangement or basis. RNA virus, on the other hand, you can see the RNA can be divided into two, icosahedral and helical only. We don't have the complex viruses for RNA viruses. They're being further categorized into naked and also envelope. After that, whether it's single-stranded or double-stranded, you can see the Baltimore classification. They have grouped the virus based on the enrollment on numbers. Okay, whether it's 1, 2, mostly they are DNA viruses. Group 3, 4, 5. Mostly they are RNA virus. So this is the Baltimore classification that we can use in order to detect or to confirm certain types of virus. And down here there are the properties. Okay, in the blue box down here, the family name of the virus. For example, the coronavirus, the virion polymerase, this negative virion diameter is 80 to 160, and also the genome size in kilobase is around 16 to 21. Okay, I'm sorry, previously it's not the mRNA. If you look at the previous one, okay, this 42, for example, 3.2 hepatna, this resembles the let me go back again, it resembles the virion diameter in nanometer and also the genome size. Okay, right, okay, check, I cannot go. Okay, we move to the helical viruses. So this is the capsid. Inside it you have the single-stranded DNA or RNA. Okay, the capsid is arranged in the helical form, and this is true in the case of Ebola virus. For the polyhedral, you can see the nucleic acid inside is being encapsulated by the capsid. You can see the capsomere there. This is on the right example of adenovirus. In the case of envelope virus, you can see the capsid inside the virus, the genome inside the capsid, and if you combine the capsid and genome, these two structures, we can call it as nucleocapsid. We have the tegumen layer. This is the floating materials inside, or the filling the space between the nucleocapsid and also excuse me, the viral capsomere. You can see the membrane glycoprotein complex. This is considered as the envelope. For the complex viruses, you can see the bacteriophage. This is a very complex structure. You have the capsid, and inside the capsid you find the DNA, and it's kind of a sheath also in between the foot and the neck. Okay, the sheath. You have the tail fiber, pinned base plates. This is very complicated viral structure. For the taxonomy, organization of organism based on presumed natural relationship. Taxonomy means how do you name them, how do you detect them or classify them based on the organization of the organism itself in the nature. Classification is the arrangement of the organism into groups, okay, based on whether it's structure, based on the DNA or genetic material, based on the genes they express, based on the characteristic, based on the shape, based on the presence of capsid, for example. However, the nomenclature is the naming. Okay, the naming, how do you give name to a certain virus? So this is just a quick definition of these terminologies. Taxonomy of viruses. Families of viruses are based on nucleic acid type. How do they replicate, and based on the morphology. That's the taxonomy of viruses. Right? How do you recognize a certain virus? Okay, by looking at the DNA or RNA, now you know that this is a certain type of virus. By looking at the type of replication, how do they replicate? Okay, is it through the lysogenic cycle or lytic cycle? Then you can know what these viruses are. Based on the morphology, the presence of capsomia or the capsid or envelope, so you can determine where does these viruses belong to. Classification, if you talk about the classification, how we can categorize the virus based on the characteristic that they have elicited. Okay, the viral morphology based on the size, shape, the length, the presence of envelope. Okay, the genome properties, DNA, RNA, size, strandedness. Okay, the DNA, whether it's circular or is it linear, the physical-chemical properties. This is the classification of the virus. Again, we have the viral protein properties. We have the genome organization and replication, which is unique to a certain type of virus. We look also at the antigenic properties, biologic properties. How do they being transmitted? What is the vectors? Are they having the intermediate vectors in between? Tissue tropism. So these are the things that makes how we want to classify the virus. What is tissue tropism? Tissue tropism or viral tropism is the ability of the virus to productively infect a particular cell. Okay, we call it cellular tropism. Okay, or if you talk about the tissue tropism, it means that the ability of the virus to productively infect the tissue, right? So this is the things, the ability of the virus to cause any considerable diseases or effect to the host cells. Nomenclature, how do we name them? If you look at the example here, family Viridae, okay, Herpes Viridae is the family, the subfamily is Virinae, for example, Beta Herpes Virinae. Genus virus, okay, we call it the Herpes simplex virus. Okay, so don't mix these things up. Okay, make sure you know which one and how do we name them. Virus families, okay, we're talking about the families virus now. Okay, the families of virus, okay, just like the Herpes Viridae and all these other viral families. Single-stranded ssDNA non-enveloped viruses, for example, Parvo Viridae, they are human viruses. They commonly infect the babies. Double-stranded DNA non-enveloped viruses, mass adenovirus. They cause the respiratory problems. The dsDNA non-enveloped. This don't have envelope. Papilloma virus that causes the human wart condition. Other than that, the genus include Polio Virus. Double-stranded DNA envelope viruses, for example, Orthopox virus, causes the vaccinia and smallpox virus. And smallpox conditions. Molluscipox virus, they cause smallpox, molluscum contagiosum, and also cowpox. Double-stranded DNA envelope virus include the simplex virus, HHV1, type 1, and HHV type 2. HHV stands for human herpes virus. Varicella virus, also HHV type 3. Lymphocryptovirus, HHV type 4. Cytomegalovirus, HHV type 5. Roseolo virus, HHV type 6. HHV type 7 don't have names so far. Kaposi sarcoma, HHV type 8, and also some herpes viruses also considered as HHV type 8. And some herpes viruses can remain latent in the host cell, means they can sleep in the host cell for some period of time, and when they want to start the effect or giving symptoms, then they will do that anytime they want. So this is just example of the structure. Double-stranded DNA envelope viruses. This includes Rna Virus. This is the Hepatitis B Virus. They use reverse transcriptase to produce DNA from mRNA. So this is a special enzyme for for the transcription process. They want to convert the double-stranded DNA into the mRNA, and the Rna virus have the capacity to use this enzyme before they're giving a certain effect to the human. In this picture, this is Hepatitis B, also a type of Rna Virus. Presented with this lady is presented with a distended abdomen due to hepatoma resulting from chronic hepatitis B infections. So can you imagine that the hepatitis B virus is very infectious and also dangerous to you? Make sure you know what you are doing and observe your cleanliness at all times. Single-stranded RNA, positive strands, non-envelope. These include the enterovirus, for example, the Polio Virus and Coxsackie Virus. Rhinovirus, Hepatitis A Virus is another type of this single-stranded positive strand non-envelope RNA virus. ssRNA, positive strand, non-envelope, includes the Hepatitis E virus, norovirus that causes the gastroenteritis. Single-stranded RNA, positive strand, envelope, this includes the alpha virus and is transmitted by arthropods. Rubivirus, rubella virus, in this case it can cause the microcephaly, PDA, and also cataracts. It is being classified under family Togaviridae. Flaviviridae is the single-stranded RNA, positive strand, and this is an envelope virus, for example arboviruses that can replicate in arthropods, causing the yellow fever, dengue, systemic lupus erythematosus, and West Nile viruses. Hepatitis B virus included in this Flaviviridae family. Single-stranded RNA, positive strand envelope, coronavirus. For example, they infect the upper respiratory tract. They are classified under family Coronaviridae. Rhabdoviridae, you can see here, for example, vesiculovirus, Lysogenic virus, Rna virus that causes from or originated from the canine or animal sources. Filoviridae is another type of RNA negative strand envelope virus that includes filovirus. They are helical in terms of the shape. For example, the Ebola. So this is an example of patient presenting with bleeding gums that causes hemorrhagic that causes by the Ebola virus, the hemorrhagic diathesis in the patient. This is a negative strand. Why some viruses are positive, having positive strand, why virus are having the negative strand. So you have to go back to the Baltimore classification, how we classify the different type of viruses based on the genome and also the replication strategy. So for example, class 1, class 2, 3 up to class 7, they have different types of positive or negative stranded. Whether it's single stranded or double stranded, they have these different types of classification. I give you example. For example, the class 1, okay, the double stranded DNA genome, example like the T4 bacteriophage, okay. Example of animal virus is the herpes virus and pox virus. Okay, herpes virus and pox virus are viruses that infect the animal. The lambda virus and also the T4 bacteriophage are the viruses that infect bacteria, but they are both considered as the class 1 double-stranded DNA genome. All right, so we have these a different class of virus based on the Baltimore classification. Let me have a look if I can have some slides to explain it further. All right, just give you an example. All right, just give you an example. For example, the single-stranded RNA that contain positive sense RNA, okay, ssRNA, positive strand, and during an infection they act like the mRNA that can be translated by the host ribosome. So it's pretty easy, straightforward if it's positive strand. But in the case of virus with the negative strand, right, they need to be converted into positive strand before the genetic material can give proteins that is important for infecting the host cells. So like in this case, like the Ebola virus, this is the example of single-stranded RNA, but they have the negative strand. It means that these viral negative strand need to be converted into positive strand by RNA polymerase, okay, by RNA polymerase to convert them into positive strand. If it's remain negative, it's quite difficult to extract the information or to translate the information into a viral protein. So they have to hijack the host mechanism system to use the RNA polymerase inside the host cell and convert into the positive strand before this positive strand virus can translate into a viral protein. Right, so this is just an example. Single-stranded RNA strand, a negative strand envelope, Paramyxoviridae. It causes the parainfluenza, mumps, and Newcastle disease. Another example is Mobile virus. You can see in the picture from the animals. Okay, it can straight away infect the humans, and from the human it can go and infect some other animals, and these circulations or this cycle continue again and again. Delta viridae, for example, Hepatitis D virus. Multiple RNA strands. Okay, it's not only single-stranded or double-stranded. I think this is you got a few strands. Okay, multiple strand, but they are negative sense. You need to convert into positive strand by RNA polymerase before producing the viral protein. Orthomyxoviridae include the influenza virus A and B, C, right? This is some of the symptoms that caused by the Orthomyxoviridae. You have the fever, sore throat, diarrhea, vomiting, coughing. So it's quite common symptoms. Multiple RNA strands, again negative strands. Bunya virus, Hunter virus. Rna Viruses is another type of multiple RNA strand with a negative sense component. 2 RNA strands produce DNA. This is the lentiviral that causes human immunodeficiency condition, or Hiv virus we call it. Oncogenic virus, because they use what we call reverse transcriptase to produce DNA from the viral genome, includes all the RNA tumor viruses. Okay, this is related to the cancers. Okay, in other words, we call retroviruses as the cancer causing viruses. Reoviridae, this is the double negative sense double-stranded RNA, non-envelope. It causes respiratory enteric orphans. Okay, another type is rotavirus that cause mild respiratory infections and gastroenteritis. Colorado tick fever is another example. Alright, so we come to these slides to differentiate between 2 types of viruses. Commonly we use this acronym HHAPPP that stands for herpes, hepatna, adenopapova, parvo, pox viruses. And most DNA viruses are double-stranded. They show icosahedron structure. They can replicate inside the nucleus, except for Parvoviridae that have a single-strand DNA. Poxviridae have a complex DNA and does not have icosahedron structure. Okay, we have 2 exceptions here. They have the 3 envelopes. Okay, for example, in the case of herpes, hepatna, and pox virus, they have 3 envelopes. Naked, they don't have any envelope, which are the papova, adeno, and also parvo. Okay, so I know it's quite difficult to get or to understand this, but I hope you can just have a look and detect what are the example of these DNA viruses. On the other hand, we have the RNA viruses, includes the toga, corona, retropicorna, calci, and so on. Okay, most of them are single-stranded. Okay, half of them are positive and half of them are negative sense. Most of them have the envelope. They show helical instead of icosahedron like the DNA virus, and they commonly replicate in the cytoplasm. Okay, unlike the DNA that replicate inside the nucleus. Alright, we also have the exception here, like the Rheoviridae. They are not single-stranded, they are double-stranded. 3 of the RNA viruses that are non-enveloped. These include Pecorna, Calici, and Rheoviridae group. We have 5 icosahedral symmetry. Okay, Rio, Pecorna, Flavi, Calici, Rhabdo, and Toga. They are not helical structure. Right, we have lots of exception here. 2 undergo replication in the nucleus. This include the retro and ortho myxome. Okay, again, this is a simpler form for you to digest. If you talk about the DNA viruses, of course, just previous time I've shown you the complex DNA viruses too, but this is just I know this is the earlier or the classical or simple what we call categorization of DNA virus, whether they are enveloped or they don't have these envelope or naked, right? So the example or the most common one has been highlighted there. In the case of RNA virus, single-stranded and double-stranded, you can see the example of the type of viruses. Good. You know that the virus can be grown inside the lab actually, and how do we want for the medical purposes, for research purposes, usually we grow the viruses to study certain diseases or to infect or to see where these viruses binds to, or what does the symptoms are being caused by these viruses. There are lots of studies or the virology studies that manipulating a certain types of viral DNA, and then they want to manipulate these for looking or to deliver certain cells or certain genetic materials inside the animals or the human tissue. Well, good to know that we can grow our own viruses, and they must be grown in living cells, for the purpose of study. For example, the bacteriophages, they form plaques on lawn of bacteria. On the E. coli, for example, you can see that they forming the plaques. And of course, the bacteria is the living cells. The animal viruses may be grown in living animals on the embryonated eggs. Animal and plant viruses may be grown in the cell culture too. Alright, so this is just for you to know how we can grow and inoculate certain materials from the embryonated egg, and then we can also introduce the viral particle in any segments of this egg. Right. On the other hand, in the lab, if you're interested in growing the virus for delivering genetic materials or to study certain lineage or the origins of cells or tissues, you can use that by what we call centrifuging the tissues with the enzymes, and after that you culture the cells in the suspended culture. After that you can isolate the virus of interest. Now we have moved on the multiplication, on how the virus can multiply or replicate inside the host system. For example, inside our body. Okay, how the virus attack our body system and how our body system react to this attack. Right? For a virus to multiply, they must invade the host cells, okay, and then take over the host metabolic machinery. They must hijack our system. If they are not strong enough, means that they can easily be eliminated. All right. Most of the cases, okay, we found that the virus attacking our body system and our body immune cells are capable in safeguarding our body, so there's no infections occur. But at some conditions, in the case our immune system is very low, the virus can attack and hijack our metabolic machinery and try to inject their genetic material and start the replication inside our cells, and after that they will come up with a huge number of viral particles or viruses that can subsequently infect our cells or our tissues or our body, or they can leave our body system to infect other people or other organisms. Alright, so we're going to look how this virus being replicated or multiplicated inside the host system. Basically, we can categorize them into two mechanisms, the lytic cycle and the lysogenic cycle. The lytic cycle ends with lysis and the death of the host cells. This is fatal. Multiplication of the T4 bacteriophage in E. coli, for example. They can stick to the surface of the E. coli and inject their DNA. As for the lysogenic cycle, they don't cause the fatality to the host cells. Instead, the cells can remain active. Alright, let's go through the lytic cycle. You see, it starts in a cascade or step-by-step process where it starts from the attachment, where the phage attached by tail fibers to the host cells, and then they penetrate, open the cell wall, okay, kind of protruding or try to poke the cell wall, and then the tail sheaths contract to force the tail core and DNA into cells. Once they inject, okay, our cells, it kind of producing a hole for them to allow the DNA material from this phage to enter the host cells. And after that, they start to produce or multiply these DNA components and certain types of proteins. This is the biosynthesis phase. After that, these DNA components start to mature, become phage particles, and they can invade some other location, or it can leave the host cells. Once the host cells is already died or being or broken down, the phage lysozyme, by the phage lysozyme enzyme, they can leave and enter some other organism. So this is an illustration how is the process begin. So this is the phage. You can see within the capsid we have the DNA, and inside the sheath, okay, you can see the tail attached to the outer surface of the E. coli. You can see inside the E. coli they have the bacterial chromosome, they have the cell wall, but the bacteriophage is capable to poke into the cell wall, and then they release the DNA material, okay, as it penetrates the host cell and inject its DNA material. You can find that they start the biosynthesis or the production of a new viruses, okay, and then they can retain there for some period of time until it's become mature, and they can be released, all right, outwards, okay, can be released to the outer environment by enzyme called lysozyme, and these new virions or viruses can infect any other peoples or any other organisms, tissue or cells. Alright, so the maturation step you can see on the circle here, start from the tail, okay, it have the sheath, and it forms the capsid. All of these are happening for some time before they become mature. Let's move on to the lysogenic cycle. On the left hand side you can see in red it's the lytic cycle, means it can cause death to the host cell. However, in the case of lysogenic cycle, in the blue color on the right, it can, the cells, the host cell can remain active, but they integrate some of the phage DNA into the bacterial chromosome by a process called recombination. All right, so they have the genetic material from the viruses. Okay, it's not only the bacterial chromosome. So of course later on they can produce a different types of E. coli that have some rearrangement or recombination event. Means that they already modify the structure or the bacterial chromosome, but the bacteria remains alive. Okay, it just some modification has been made. The lysogenic cycle can give rise into three things, okay, three events. The lysogenic cells are immune to reinfection by the same phage. It means that the second time the phage comes to invade the bacteria, it's become immune now. Okay, so it cannot cause any fatality or any undesirable effect. The second event happening is the phage conversion. It means that the host cell exhibits a new properties, maybe a new strain, a new E. coli strain as compared to the first one that is not infected by the virus. The third event is the specialized transduction. It's mediated by lysogenic phage that package the bacterial DNA along with its own DNA in the same capsid, but only certain genes can be transferred. And this is very unique because they're selecting only a certain genetic locus to be transferred. Okay, not the whole the whole DNA sequence. So this is an example. You have the E. coli and together with its bacterial chromosome or the bacterial DNA. You can see here the prophage is already integrating their DNA component. Alright, so you have three things here. You have the galactosidase gene here as a part of the bacterial DNA, and you can see the phage inserting their DNA inside it too. Okay, what is prophage? Prophage is the genetic material of a bacteriophage. It's incorporated into the genome of bacterium and able to produce phages if specifically activated. Right? So now we move on to the next stage where the phage genome excises. Okay, you can see it excises here, but they carry the GAL gene. Alright? They cut from the first one and bring inside the phage DNA, leaving the bacterial DNA alone in the circular shape in purple. And after that, this GAL gene plus the phage DNA being inserted into the capsid of the bacteriophage at the maturation stage. Okay, so this capsid or the bacteriophage that contain the GAL gene, a specific gene, can be inserted or into another E. coli. Okay, and then start the recombination process. So this is only allow a certain set of gene of interest to be transferred into a next organism. Okay, like the previous one, you know that the whole bacterial chromosome or the whole bacterial DNA being transferred, but in this specialized transductions, only certain genes is being transferred. Multiplication of animal viruses again is quite similar because it involves the attachment of viruses to the cell wall membrane of the host cells, penetrates by endocytosis or fusion uncoating by viral or host enzymes, means they release. After that, the biosynthesis or production of new nucleic acid and associated protein. After that, maturations, where the nucleic acid and capsid protein, they assemble together to make the mature, fully formed bacteriophage, where they can be released to the outer environment. So this is the illustration to illustrate the process. It begins with the attachment of plasma membrane to the host cells. You can see it's encapsulated into a form of vesicle, and it enters the cytoplasm to a process called endocytosis, where they start to penetrate, and then they will release the coats and leaving the DNA materials inside the cytoplasm. Biosynthesis of DNA viruses. DNA containing viruses, they can replicate their DNA in the nucleus of the host cell by using the viral enzyme. They have their own enzyme to help them to replicate inside any host cells, and then they synthesize their capsid and other proteins in the cytoplasm by using the host cell's enzyme. So let's have a look at the multiplication of DNA virus. It starts with the virions. Okay, this is example of Parvo Virus, which is the DNA virus. They contain capsid, and inside the capsid they have the DNA in a circular form, and the virions start to attach to the host cells. Alright, this is the DNA in purple and cytoplasm of the host cells, and then the virion penetrates the cell and its DNA is uncoated. When the DNA is uncoated, it's quite easy for them to invade the nucleus. Okay, once they invade the nucleus, they can disrupt or they can disturb the process of transcription and translations that is already happening inside the host cells. They try to hijack the machinery, okay, by introducing their own DNA particles. And you can see at the number 4 and number 5 that the capsid protein start to be synthesized, and the virions get mature before they become available to be released out from the host mechanism, out of the host cells. Now leaving the host cells where they can still survive, but most of the time they can be fatal, okay, or died immediately. In terms of the biosynthesis of the RNA viruses, there are several different mechanisms of mRNA formation occur among different groups of RNA viruses. You can see here on the left hand side is a viral nucleic acid, the middle the virus family, and on the right hand side the special features of biosynthesis. I take example of the RNA viruses with the positive sense strand. For example, Rna Viruses family and Toga Viridae. What are the special features? The viral RNA functions as the template for synthesis of RNA polymerase, which copies negative strand RNA to make mRNA in cytoplasm. RNA negative strand, on the other hand, for example, Rhabdoviridae, the viral enzyme copies viral RNA to make mRNA in the cytoplasm. RNA double negative stranded Rheoviridae, for example, the viral enzyme copies negative strand RNA to make mRNA in cytoplasm. For the complex, I think, the RNA reverse transcriptase is another enzyme that can facilitate this conversion. Viral enzyme copies viral RNA to make DNA in cytoplasm, and then DNA moves to the nucleus. So this is at your level, I think this is not that important, but it's enough for you to know the virus family and also the type of viral nucleic acid. This is an example of the pathway of multiplication for RNA containing viruses. Of course, it's a bit busy structure. You can see the what you call the strong blue color here is the viral genome. The middle one is the positive sense strand viral genome. The pale one is the negative or antisense, we call it, a strand of the viral genome. SS is a single-stranded, DS is double-stranded. It starts from the attachment to the host cells, again just like the DNA virus. After that, they penetrate and uncoating all the proteins, releasing the genetic material. If it's a negative strand, okay, if it's negative sense strand, they need to be converted into positive strand first. Okay, if it's already positive, it can be directly translated into the proteins that makes the virus. This can include the capsid. It can include some other proteins particles. After that, it can be matured and released out from the environment. Okay, if it's if the strand is positive, okay, it must first be transcribed from the negative viral genome before protein can be synthesized. Okay, it's another way around. If it's already positive, it's fine. You just continue with the translation to make a mature virus. Alright, so this is C in the case of double-stranded RNA, whether it's positive or negative sense. In the case, for example, Rna Viruses, okay, mRNA is produced inside the capsid and released into the cytoplasm of the host cells before capsid protein be produced and continue with the maturation. So in the exam, if I ask you, give me an example of single strand RNA or which is positive strand, you can say that it's a Rna Viruses family. For single strand RNA negative sense, Rhabdoviridis. For double stranded RNA positive or positive sense, you can say it's the Rna Viruses. Virus and cancers. Okay, the cancers progression and initiation most of the time being triggered by the viruses that causes mutation in certain DNA strand of the human cells. So the transformation of the normal cells into the tumor cells most commonly being triggered or stimulated by the viruses. And most of the time, if you look back in your pathology class, if you look back in your biology classes or biochemistry class, you can find that the oncogenes, okay, these are the set of genes that cancer causing genes. Okay, these are the genes that produce or makes the cancers inside the cells or tissue. On the other hand, you can see that the oncogenic viruses can also trigger this tumor condition in humans or in animals. Okay, even though it's without the help of oncogenes, the virus can interfere and causes the tumor too. Okay, when the tumor cells undergo transformation, they acquire properties that are distinct from the properties of uninfected cells or from infected cells that do not form tumors. Antigens acquired are these tumor specific transplantation antigen on their cell surface. This is a specific antigen that is expressed or acquired during this process. The T antigens present in their nucleus. Some distinction between two types of oncogenic viruses. The oncogenic DNA viruses includes Adenovirus, Herpes Virus, Pox Virus, Papilloma Virus, and Rna Viruses. Whereas the oncogenic RNA viruses include Retroviridae, which is the viral RNA is transcribed to DNA, which can integrate into host DNA, and also HTLV. HTLV is the human T lymphotrophic virus type 1. Okay, or some books they call it as human T cell leukemia virus type 1. The virus can cause a type of cancer. We call it adult T cell leukemia or adult T cell lymphoma or ATL. This HTLV type 1 is transmitted primarily through infected bodily fluids that includes the blood, breast milk, and also the semen, right? Through the fluid or the bodily fluid. Viruses and cancer, again, we have the DNA oncogenic viruses. They can be found within several families of DNA containing viruses like the Epstein-Barr virus, excuse me for that, or EBV. They cause infectious mononucleosis, Burkitt's lymphoma, and nasopharyngeal carcinoma or cancer. Hepatitis B virus, liver carcinoma, 98% higher in people with prior to hepatitis B viral infection. Okay, this is just an example of this Burkitt's lymphoma. Viruses and cancer again, but for RNA oncogenic viruses, only the oncoviruses in the family Retroviridae cause cancer. So this is important. Not all RNA viruses cause cancers. Okay, we have a few exceptions. Only oncovirus in the family Retroviridae can cause cancers. Okay, for example, the HTLV type 1 and HTLV type 2. The ability of these retrovirus to induce tumor is related to their production of reverse transcriptase. Some contains oncogenes and some other contain promoter of oncogenes. All right, so I hope you understand about that. Let's move on to the latent viral infections. What does the latent means? Latent means a virus can remain in equilibrium within the host and not actually produce disease for a long period of time. Most remain in the nerves, wait for decreased host resistance, then perform a sudden attack. Okay, so it's kind of sleeping, latent, so you didn't know that. Okay, it can be in our body right now. Okay, but as we live like normally, they can come back. Okay, they can, become activated and perform a sudden attack. Okay, for example, herpes viruses, herpes simplex viruses, chickenpox viruses, varicella zoster that cause shingles, they are all classified under latent viral infection. So this is example of shingles. They can remain in your body for a long period of time. In contrast, we have the persistent viral infections, that a disease process that occurs gradually over a long period. It always persists, always consistent over a long period of time. It always come and go and then come again and go again. So, it's never-ending process. So it is true for measles virus that causes for a rare form of encephalitis. We call it subacute sclerosing panencephalitis. It's detectable infectious virus gradually builds up over a long period of time rather than appearing suddenly. Okay, it gradually builds up, okay, and then give an attack, and after that keep dormant for some time and then come back. Okay, it's a never-ending process too. All right, prions. Why it is called prions? It's a type of it's a disease. Okay, It's taken from the name proteinaceous infectious particles. Okay, we call it the prions. Okay, with the proteins that cause this mad cow disease. Okay, and the transmission can be genetic, means it can be inherited from the parents to the offspring, or from feeding scrappy, or from eating from these animals products that already these prion proteins already present inside this animal product, this infected meat, okay, or in infected cow's meat. This is important for us to avoid eating undercooked beef from infected cattle, right? So it's quite difficult for us to detect, to make sure whether our meat we are consuming is free from any infectious material. CJD, the Creutzfeldt-Jakob disease, has been transmitted with transplanted nerve tissues and contaminated surgical instrument. Okay, it's in the hospital settings, for example, where the contaminated instrumentation is being infected, and people keep on sharing, and cross contamination occur, and it can cause this type of disease. Plant viruses, they can enter through wound or via insect bite. For various infectious to RNA, okay, potato spindle tuber disease can infect the human too. These are the list of medically important viruses. I classified them in yellow if you want to have a look in the next slides. Okay, these are the most commonly discussed viruses in the clinical setting. The most, or the top one, is the herpes virus. It can be type 1, type 2. It can be from the herpes family, varicella zoster or Epstein-Barr virus. They are all from the same family. They cause latent infection. It can be reactivated by immunosuppression or previous infection. How do you diagnose them? We use the zinc smear. If you see a positive intranuclear inclusion or multinucleated epithelial giant cell, which is positive staining, so you can conclude that this is positive for a certain herpes viruses. What are the diseases associated with HSV? It include the herpes labialis, gingivostomatitis, conjunctivitis, keratitis, and encephalitis. Mode of transmission, it can be direct contact or via saliva. Herpes simplex type 2, it can cause genital herpes, neonatal herpes, meningitis. It can be transmitted through the sexual contact, passage through birth canal. Varicella zoster, for example, this is the most common one where everyone got it. Chickenpox, which is the primary infection. The secondary infection of varicella zoster, we call it shingles or herpes zoster. It can be transmitted through respiratory droplets. Its incubation period is within 2 to 3 weeks. Primary infections confer partial immunity. Okay, you need to have that partial immunity. Latent infection reside in the dorsal root ganglia. Of course, it's involving the CNS. Reactivation is because of stress, sunlight. This triggers the reactivation of VZV. So you can have, this is quite similar to what we have discussed previously. Just for suppressing them, you can use the antiviral regimens that include the acyclovir or valacyclovir. Herpes viruses, you can see the mode of transmission where it's transmitted through the respiratory route. It can stay latent in your body system, and after that they can specifically bind to the nerve cells in the dorsal root ganglion. That is invading the a central nervous system via this blood. I think not blood, via the peripheral nerve. Right? Shingles, almost the same, but when you are being infected or the viral DNA is already inside the host cells, they will release all these viruses or the mature viruses, and they move down to the peripheral nerve, okay, and attack the spinal cord. All right. So reactivation of the latent HHV type 3 releases viruses that move along the peripheral nerve to the skin. This is just example where we start with the vaccine production in 1963, and after that being used for more than 20 to 30 years before it's completely being eradicated from our population. Epstein virus, the most common diseases associated are mononucleosis and Burkitt's lymphoma. Can be transmitted through the saliva. It enters the pharyngeal epithelium, invade the cervical lymph nodes, which is the B cells, and later the spleen or the liver. Hepatitis B virus, it's due to the exposure to the blood or the bodily fluids. It can cause lots of complications such as the jaundice. You can have these cirrhosis and also the hepatocellular carcinoma. This is another type of HHV type 4. HHV type 5, like a cytomegalovirus or CMV viruses. Second type, the commonly known viruses in hospital or in medical settings, we have Rna Non-Encapsulated Virus. The top one is the Adenovirus that causes pharyngitis, pneumonia, gastroenteritis. It can be transmitted through the respiratory droplets. If you're sneezing, if you have any, droplets coming out from your nasal root or from your, respiratory roots, it can immediately infect other people. How we can diagnose them? Through the fluorescence antibody test and complement fixation tests. Papilloma virus, it can cause the cancers of the cervix. This is important for the female patient. Mode of transmission, it can be direct contact or through the sexual activity. It has a tropism, or it has the ability to invade the squamous cells and causes the warts. Okay, the warts are the small, the small bulging conditions on the skin, okay, on the squamous epithelial linings, and this can be resolved within years. And pap smear is one of the most common diagnosis to detect this papilloma virus. The genital warts, as I have mentioned earlier, if you want to get rid of them, we can use the interferon regimen to treat these viruses. Also, we can use the DNA test to detect the type of these viruses, DNA, and also you are also required to go for the HPV vaccination so that your body is protected or have the antibodies to fight against the papilloma viruses. Smallpox viruses can cause the smallpox conditions. Also, it's being transmitted by the respiratory routes. The third one is the RNA envelope virus. This includes the influenza virus. Lately, we been bombarded by lots of different notions of viruses. Okay, what are these influenza viruses doing? Okay, the influenza viruses, they has the ability to cause the epidemics, or to certain extent pandemics, due to the antigenic drift or antigenic shift. What is antigenic shift or antigenic drift? The antigenic drift refers to the gradual accumulation of point mutation during annual circulation of influenza as a consequence of high error rates associated with RNA-dependent RNA polymerase during viral replication. It's quite long, quite lengthy definition, but it just to let you know that the antigenic drift means the same virus can have a different mutation, and it can render the type of glycoproteins that surrounded the outer layers. Okay, it can give rise to a different variants. For example, you have influenza A due to the antigenic drift, okay, or it's due to evolution or due to mutation, it can be converted into influenza B. All right, so this is quite dangerous and it's quite lethal because the transmissibility of them is being manipulated or being modified according to the environmental changes. The capsules of these influenza virus contain HA, which is the hemagglutinin and neuraminidase. Hemagglutinin has tropism for salicylic acid, which is present in the cell membrane of red blood cells and respiratory tract epithelium. This is needed for absorption. So as I said, they have a special glue for this component to go and bind to the a cell or to the tissue. Okay, in this case it's to the cellular membrane of the RBC. So this virus is easy for them. Okay, it's easy for the viruses, for the influenza virus to bind to the red blood cells and carry this viral particle through the circulation system, and this is also can give a detrimental effect to the body. Na, which is another enzyme, hydrolyze the neuraminic acid, which is a component of mucin. All right, so it hydrolyzes the neuraminic acid, which is a component of mucin that is present in the respiratory tract. Again, this kind of, killing the two birds with one stone. These influenza virus have HA and Na particles that can, bind to whatever surface they want in the respiratory tract or inside the blood. All right, only type A, B, and C can infect human. The outcome is that we can have flu and pneumonia. The symptoms and signs include fever, chills, headache, myalgia, muscle pain, arthralgia, joint pain that can be resolved within 3 to 7 days. You can have the secondary bacterial pneumonia. All right, again, the respiratory, of course, it's from the respiratory route. How to get rid of these influenza virus? You can take the vaccination, or you can be prescribed with amantadine. Pox virus causing the smallpox or variola. Okay, it caused by the orthopox. Variola major has a 20% mortality. Variola minor has less than 1% mortality. Okay, another 80% or 79 or 80% and below means that they are survived if we are being infected by the pox virus. Influenza, okay, already discussed about that. So this is just to illustrate the structure. We have the hemagglutinin spike, we have the neuraminidase spike. This is the attachment site for the binding. All right. The antigenic drift or antigenic shift is two different terminology. Antigenic shift means changes in H and N spikes, probably due to genetic recombination between different strains infecting the same cell. Antigenic drift refers to the mutation in genes coding H or N spikes, involve only one amino acid, and allow virus to avoid mucosal immunoglobulin A antibodies. Alright, so this is two different terminology. Influenza serotypes, you can find them in H3 and 2, H5 and 1, H1 and 1, H2 and 2, and some other serotypes. This is a different, you can call it a different types of influenza viruses. Coronavirus, they are envelope viruses with a positive sense, single-stranded, and they are very quickly to be transferred or to infect other organism. They have a nucleocapsid in the helical symmetry, but you cannot see its helical properly here. It can cause disease in mammals and birds. It can cause, respiratory problems again. Diseases like COVID-19, you can find the MERS-CoV and also SARS-CoV are the result of these coronaviruses. How about the latest one, the Omicron? I think is important for you to know the differences between the Delta coronavirus, the Omicron one. What are the differences, how they can cause the disease? Are they more lethal? Did Omicron more lethal, more easily transferred as compared to the Delta virus? Please let me know, because you will have a question in the final exam discussing about this. Transmission and life cycle of SARS-CoV-2. You can see the SARS-CoV-2 have a circular structure, and of course they have these set of protein, and the most important one or the most popular one is the spike protein. The spike protein binds to the ACE receptors that is available inside the respiratory tract, like lungs, like your trachea. And once the virus enters through the oral route, once you inhale some of these respiratory droplets, they can bind to the ACE2 receptors, and after that they will release all these viral particles, which is in the form of RNA, and they will start to produce their a new viral particle and encapsulate themselves before they being released out from the host cells. And after that, these viruses are being ingested by our immune system, called the antigen presenting cell in yellow, with a very with lots of food processes like a food. And these APC cells will present all the information that they receive from these SARS-CoV-2 viruses to be translated by the T helper cells. So the T helper cells is the decision maker, whether they want to disseminate or spread the information regarding the invasion of this new SARS-CoV-2 virus to the B cell or to the cytotoxic T cell. If the information is presented to the T cells, it can be immediately destroyed. Right? If your immune system is intact and you have very high immune system, you can get rid or you can destroy this SARS-CoV-2 by your own. In other hand, in other case, you can use the B cell mechanism or machinery to produce the plasma cells. The plasma cells can then generate a specific antibody for a long-term infection removal. Okay, for long-term SARS-CoV-2 removal. So for the next time when you have a second infection, the body just produces the same antibody that detects or that is capable to get rid of the SARS-CoV-2 viruses. Some differences, I have put it down here based on the structure. What are the differences between influenza virus and SARS-CoV-2? Maybe you want to learn more and read more about the florona, how people can get rid of them. Alright, so let me try to go back to the next slides. Measles viruses, mumps viruses, rubella. So this is MMR mostly being introduced, or this vaccine being introduced to the kids once they were born. Okay, maybe in the first 6 months and first year of the baby. Maybe this is the compulsory vaccination to prevent the infections of the said viruses. Okay, this is mumps and rubella. All right. HIV viruses, it can cause AIDS. It's due to the exchange of bodily fluids via sexual activities, alright, via transplant placenta, okay, after the delivery or post delivery of the baby. We can diagnose them based on the screening test using the protein adsorbance or the enzyme link immunosorbent assay. We call it ELISA. Western blotting, we can confirm the size of the protein that has been translated or being synthesized. Hepatitis C viruses can cause the hepatocellular carcinoma or chronic hepatitis. It's most commonly been transmitted via blood transfusion. People can get it from the sexual contact. Alright, rabies virus, we have discussed this, and in fact you already learned in your PBL session how this Rhabdoviridae family causing rabies encephalitis can promote or can propagate the virus to infect the central nervous system. Right? Rhabdoviruses, of course, this is one of the example that we already discussed in detail in your PBL session. And finally, the RNA non-encapsulated viruses. This includes polio viruses that cause meningitis, that leads to the paralysis and atrophy. Coxsackie virus, causing pericarditis, myocarditis, or meningitis, both lungs and also the brain. It's been transmitted through the fecal oral routes. Polio myelitis is caused polio virus. Recently, I think in 2016, or I think last year, we have a case of polio again. We think, or we thought, that the polio is fully eradicated from the society, but it's striking us back. So we make sure we have the vaccines too. It's quite important to prevent us from getting the polio disease again. You can see from the line graph here, we completely eradicating the polio since 1998, but after, I think, after 20 to 30 years. All right, last year we have a new cases. I think around 100 to 200 new cases have been reported. Rotavirus, this is the most common virus, belong to the Rheoviridae family. It causes the gastroenteritis, inflammation of the stomach. Rhinovirus, it causes the common cold, and hepatitis A virus causing the hepatitis. This is some of the explanation regarding these viruses. All right. So I think this is in some countries like in the US, they already map this encephalitis in the society, and of course they are being transmitted by a certain type of vectors. It can be Aedes, Culex, or Quilizeta that brings all these viruses and homing into the birds or in the horses on some other animals, and it can possible to infect in some other animals too, or become making some other animals as a reservoir for them to mature and for them to synthesize a new particles. All right. Pneumonia, viral pneumonia as a complication of influenza, measles, and chickenpox. The viral etiology suspected if no cause determined. Respiratory syncytial virus (RSV) is common in the infants. 4,500 deaths annually. I think it's increasing according to the years. Causes cell fusion. We call it syncytium in the cell culture. Symptoms like the coughing, but you cannot know this is the RSV until you do the serologic test and antibody test from the baby's blood, and you can get rid of them by taking the ribavirin. Hepatitis, so already know about this, and I already listed the transmission, what are the causative agent, and whether they are chronic or not, and whether the vaccine is available or not. So this is the example of the Hepatitis B virus. You can see it's quite elongated virus. Okay, this is the filamentous particle. They have the tubular envelope particles. They have spherical particles too. So there are lots of different types of shapes. If you go to the lab, if you want to do the diagnosis, maybe these are the common things. You check for the antibody. You check whether they can grow into the tissue culture. You can check whether they can endocytose and causing syncytium. You can check based on the absorbance level. You can check the protein formation through the Western blotting and checking the size of the protein to confirm whether they are the proteins being produced or originated from the virus or from the bacteria or from some other antigens. We can do the polymerase chain reaction. We can multiply lots of these DNA materials. We can multiply them before we determine what are the the sequence of interest. We can have the serology test to check the antigen antibody bindings, whether they are increasing in certain types of immunoglobulins. All right, I think that's it for today. I know it's quite fast with a lot of information, but I'm sure you'll be able to digest, even though it's not full, but at least you can understand half of it. But if you got any question or ambiguities that you think is worth to address, if there's some wrong information that you have found inside the slide, kindly let me know by emailing me to the email that I have provided here. But let me try to share with you guys again. Please contact me to this email address. I think if you want to see the lecture, you can visit my YouTube channel there. I think that's it for today. I hope you can answer your exam question, and if you got any question, you can text me, you can WhatsApp or whatever platform you want. You can reach me, and we can have we can have a revision class for you to understand further, and maybe we can discuss about the question that might, I can give a hint, the question that can come out in your final examination. If not all, but maybe just for the sake of discussion, it can take around maybe less than half an hour. Okay, but you can discuss with your batch leader or group leader. I can only have one class for that session. All right. Thank you very much for your attention. Please don't forget to sign in your attendance. The link can be found in my personal web page. You can type in Faris. I think you can. Let me try to put it down here. Not here. Let me check if. Let me try to change. Yes, maybe I can change a bit here. Maybe you can go to Faris farislab.myportfolio.com/ I think you can go to biochemistry. Okay, this is for the attendance. All right, for section 1, 2 and 3. Okay everyone, thank you very much. I'll see you again. Bye bye. We close the session by tasbih kafarah and suratul Nas.